There is a plant growing in the cracks of your sidewalk that contains more omega-3 fatty acids than fish oil. Not similar amounts, more. And for the last 70 years, chemical companies have spent billions convincing you to poison it. This is Portulaca oleracea. The Greeks called it andrachne. The Romans called it Portulaca. Every Mediterranean civilization, for 4,000 years, cultivated this plant as a staple crop, not as medicine, as food, primary food. Then, in 1947, something changed. The plant you call purslane has red stems, thick jade-colored leaves, and a sprawling growth pattern that allows it to colonize disturbed soil faster than almost any species on Earth. It thrives in heat. It laughs at drought. A single plant produces 52,000 seeds. Those seeds remain viable in soil for 40 years. This is not fragility. This is biological dominance. In 2014, researchers at the University of Maryland analyzed the fatty acid profile of Portulaca oleracea using gas chromatography. They found that 100 grams of fresh purslane leaves contained 400 milligrams of alpha-linolenic acid. That is the highest concentration of omega-3 fatty acids ever recorded in a leafy vegetable. For context, spinach contains 12 milligrams per 100 grams. Arugula contains 170. Purslane contains more than twice the omega-3s of arugula and 33 times more than spinach. But the chemical companies that manufacture glyphosate, Roundup, list Portulaca oleracea as target weed number 7 in their agricultural guidelines. They recommend pre-emergent herbicides containing pendimethalin, a chemical that disrupts cell division in germinating seeds. The application rate is 3.8 pounds per acre. Every spring, American homeowners spray 80 million pounds of synthetic herbicides on their lawns. Purslane is listed as a primary target on the back of every bottle. We are paying to exterminate the most nutritious plant in our ecosystem. Let me take you to Thebes, 1400 BC. Egyptian physicians recorded medical texts on papyrus scrolls that survive today in museums across Europe. The Ebers Papyrus, discovered in Luxor in 1862, is 110 pages long. It contains 700 medicinal formulas. Purslane appears in 8 of them. The Egyptians used purslane to treat intestinal parasites, liver inflammation, and shortness of breath. They prescribed it raw, boiled in wine, or ground into a paste with honey. The hieroglyph for purslane appears in tomb paintings showing garden plots beside the Nile. This was not wilderness foraging. This was agriculture. In ancient Greece, Theophrastus wrote Enquiry into Plants around 300 BC. He classified purslane as one of the 5 essential pot herbs, alongside beet, mallow, auric, and chicory. He noted that purslane stayed crisp in summer heat, while lettuce wilted. Greek soldiers carried purslane in their field rations during Alexander's campaigns into Persia. The plant followed Roman legions across Europe. Pliny the Elder, writing in 79 AD, recommended purslane for 41 separate medical conditions. He called it refrigerans herba, the cooling herb, because eating it lowered body temperature during fever. Archaeological digs in Pompeii uncovered carbonized seeds of Portulaca oleracea in multiple households. The seeds were stored in ceramic jars alongside wheat and barley. Food, staple food. When Islam spread across North Africa and into Spain between 700 and 1200 AD, purslane traveled with it. Arabic physicians incorporated it into Unani medicine. The plant's Arabic name, rijla, appears in medical texts from Baghdad to Cordoba. Ibn Sina, known in the West as Avicenna, prescribed purslane juice for bleeding gums and ulcerated intestines. By the medieval period, every monastery garden in Europe cultivated purslane in raised beds. Hildegard of Bingen wrote in 1150 that purslane calms excessive heat in the body. Charlemagne ordered purslane grown in every imperial garden under his 812 AD decree, the Capitulare Divii. Then, the plant crossed the Atlantic. By 1800, purslane grew in every American kitchen garden, from Massachusetts to Louisiana. Thomas Jefferson planted purslane at Monticello. Martha Washington recorded a purslane pickle recipe in 1749. American farmers planted it intentionally, beside tomatoes and cucumbers. And then the herbicides arrived. In 1947, the U.S. Department of Agriculture began promoting synthetic herbicides to American farmers. The chemical was 2,4-D, originally developed as a biological weapon during World War II.
Dow Selective Weed Killer, Dow Contact Weed Killer, 2,4-Dow Weed Killers, and Esteron 44. One of the newest and most versatile weapons in Dow's arsenal of chemical warfare is 2,4-Dow Weed Killer, proved in action against hundreds of weeds in a wide variety of crops. 2,4-D kills the entire weed, roots and all. Its victims include most broad-leaved annual weeds as well as perennials. Even perennials with deep root systems may be eradicated by repeated applications. Most grasses are not harmed when it's properly applied, nor is the soil poisoned. This time-lapse photography shows in a few moments the gradual killing of a common weed over a 15-day period after spraying with 2,4-D weed killer. 2,4-D is a growth regulator type of weed killer. The action is physiological, appearing to upset the plant's growth processes, causing modification and breakdown of its parts, and causing gradual but certain death.
The war ended before deployment, but chemical manufacturers had production facilities running at full capacity. They needed a peacetime market. The market they created was weeds. But here is the problem. In 1947, farmers did not consider purslane a weed. They considered it food. The shift required propaganda. Chemical companies funded agricultural extension offices to publish pamphlets identifying, quote, problem plants in crop fields and lawns. Purslane appeared on every list. By 1960, the herbicide market in America reached $100 million annually. Purslane was reclassified from vegetable to pest in USDA publications. Seed catalogs stopped listing it. Agricultural colleges stopped teaching cultivation methods. The plant that every civilization for 4 millennia had deliberately grown was now something to exterminate. In 1974, Monsanto introduced glyphosate under the trade name Roundup. The original patent lists 39 target species. Portulaca oleracea is number 12. The suppression accelerated. Home improvement stores began selling, quote, weed and feed lawn treatments containing dicamba, an herbicide that kills broadleaf plants. Application instructions specifically mention purslane. The chemical label lists lawn beautification as the primary benefit. Translation: a monoculture of one grass species is beautiful. Diversity is ugly. By 1990, glyphosate sales exceeded $1 billion annually. Monsanto released Roundup Ready crops, genetically modified plants immune to glyphosate, allowing farmers to spray entire fields without harming corn or soybeans. Every other plant dies, including purslane. In 2020, global glyphosate sales reached $9.2 billion. And during this 70-year chemical campaign, something extraordinary was happening in laboratories. In 1986, Dr. Artemis Simopoulos, a physician and nutritionist, analyzed the fatty acid composition of wild purslane growing in suburban Maryland. She used lipid extraction and high-performance liquid chromatography. The results stunned her. Purslane contained 400 milligrams of alpha-linolenic acid per 100 grams of fresh leaves, but it also contained 12.2 milligrams of eicosapentaenoic acid, EPA, per 100 grams. EPA is the omega-3 fatty acid found in fish oil. It does not exist in land plants, except purslane. Simopoulos published her findings in the American Journal of Clinical Nutrition. The paper received little attention. She was reporting nutritional data on a plant that American agriculture had spent 40 years teaching people to kill. But other researchers began investigating. In 1992, scientists at the Agricultural University of Athens measured the vitamin content of Portulaca oleracea. They found 1320 micrograms of beta-carotene per 100 grams. Carrots contain 835. They found 21 milligrams of vitamin C. Oranges contain 53, but you eat purslane raw, with no loss from cooking. In 1998, researchers in Mexico identified 14 different flavonoids in purslane extracts. Flavonoids are polyphenolic compounds that function as antioxidants. The two most abundant were camphorol and quercetin. Quercetin inhibits histamine release, which explains why traditional medicine used purslane for allergic reactions. In 2002, Chinese researchers isolated a polysaccharide from purslane called POP1. They tested it on diabetic mice. Blood glucose dropped by 41% after 4 weeks. The mechanism involved increased insulin sensitivity in liver cells. In 2009, a team at Cairo University fed purslane extract to rats with chemically induced liver damage. After 30 days, liver enzyme levels normalized. Histological examination showed regenerated hepatocytes. The active compound was identified as dopamine. Yes, the neurotransmitter, at concentrations of 16 milligrams per 100 grams of fresh plant. In 2014, Iranian researchers tested purslane mucilage on human stomach ulcers. The mucilage formed a protective coating over ulcerated tissue, reducing healing time by 60% compared to controls. The mechanism was identical to modern pharmaceutical treatments, but without side effects. The scientific validation continued to accumulate. Every study confirmed what ancient civilizations knew empirically. But here is what none of those studies explained. Why does purslane produce omega-3 fatty acids when no other common vegetable does? The answer lies in photosynthesis. Most plants use C3 photosynthesis, the standard method of fixing carbon dioxide into glucose. But purslane uses CAM photosynthesis, crassulacean acid metabolism. This is a survival adaptation for arid environments. Standard photosynthesis requires plants to open their stomata, leaf pores, during daylight to absorb CO2, but open stomata leak water. In hot, dry climates, this causes fatal dehydration. CAM photosynthesis solves this problem. Purslane opens its stomata at night, when temperatures drop and humidity rises. The plant absorbs CO2 in darkness and stores it as malic acid in vacuoles. At dawn, it closes its stomata completely. During daylight, it converts the stored malic acid back into CO2 inside the sealed leaf and completes photosynthesis with zero water loss. This is biological genius. But CAM photosynthesis has a side effect. The biochemical pathways that manage carbon storage require specific fatty acids as enzyme cofactors. To maintain membrane fluidity in leaves that undergo radical pH shifts, the plant synthesizes high concentrations of polyunsaturated fatty acid, specifically alpha-linolenic acid and its derivatives. Purslane produces omega-3s not as nutrition for animals, but as a mechanical requirement for surviving drought. We benefit from its survival mechanism. And here is the critical detail that ties this to human health. The ratio of omega-6 to omega-3 fatty acids in purslane is 0.5 to 1. For every gram of omega-6, there are 2 grams of omega-3. The modern American diet has an omega-6 to omega-3 ratio of 16 to 1. This ratio causes chronic inflammation, which underlies heart disease, diabetes, arthritis, and Alzheimer's. Every major degenerative disease of modern civilization correlates with omega-6 dominance. Purslane reverses that ratio. In 2004, Dr. Norman Salem at the National Institutes of Health calculated that a daily serving of 120 grams of fresh purslane provides sufficient omega-3s to match the cardiovascular benefits of weekly fish consumption. He noted that traditional Mediterranean populations who ate purslane regularly had 70% lower rates of heart disease than Americans, even when controlling for other dietary factors. The plant growing in your driveway can prevent the leading cause of death in Western nations. And every spring we spray it with carcinogens. Let me tell you how to identify this plant, because if you walk outside right now, it is almost certainly within 50 feet of your door. Purslane has a prostrate growth habit, meaning it sprawls horizontally rather than growing upward. The stems are smooth, round, and reddish-pink to burgundy in color. If you snap a stem, it exudes a clear, slightly viscous sap. Not milky, clear. The leaves are paddle-shaped, about 1 inch long, thick and succulent. They grow alternately along the stem, though they often cluster at stem tips. The leaf surface is smooth, waxy, and jade green with a hint of yellow-green toward the center vein. In summer, purslane produces small yellow flowers with 5 petals. The flowers open only in direct sunlight between 10 a.m. and 4 p.m. They last one day. Each flower produces a seed capsule shaped like a tiny urn with a removable lid. When mature, the lid pops off, releasing hundreds of black seeds the size of poppy seeds. The entire plant is edible: leaves, stems, flowers, and seeds. Here is the critical identification feature that separates purslane from toxic lookalikes. When you break a purslane stem, the interior is hollow. If the stem is solid, it is not purslane. Spurge, a common toxic weed, has milky sap and solid stems. Purslane has clear sap and hollow stems. Taste test. Place a small leaf on your tongue. Purslane tastes lemony tart with a cucumber-like freshness. The texture is crisp and juicy, not fibrous. If it tastes bitter or burns your mouth, spit it out. It is not purslane. Now, where do you find it? Purslane colonizes disturbed soil with full sun exposure. Look in gravel driveways, sidewalk cracks, garden beds, the edges of parking lots, and any bare soil that receives 6 hours of sunlight. It appears in late spring, when soil temperature exceeds 60 degrees Fahrenheit and continues growing until the first frost. In northern climates, purslane emerges in May. In the south, it appears in March. The plant is an annual, meaning it dies each winter, but the seeds germinate prolifically each spring. If you want to cultivate purslane intentionally, do nothing. Stop spraying. Stop pulling. Within 2 seasons, purslane will establish itself in any sunny garden bed. You can accelerate this by scattering seeds from mature plants in July, or purchasing seeds from companies that sell heirloom vegetables. Yes, seed companies now sell purslane again, marketed as a specialty green. The irony is complete. We are buying seeds for a plant we spent 3 generations exterminating. How do you eat purslane? Fresh, primarily. The omega-3 fatty acids degrade when heated above 150 degrees Fahrenheit, so raw applications preserve maximum nutrition. Greek horiatiki salad traditionally includes purslane alongside tomatoes, cucumber, onion, and feta. The tart flavor balances the salt of the cheese. Lebanese fattoush salad uses purslane as a base green, dressed with lemon juice and sumac. You can also add purslane to smoothies. The mucilaginous texture, similar to okra, creates a creamy consistency without dairy. Blend purslane leaves with mango, lime, and coconut water. The tart flavor complements tropical fruit. Here is what matters. You can begin eating purslane today, not next week after you buy supplements, not next month after you research more. Today, walk outside. Identify the plant, wash it, eat it. No purchase necessary, no prescription required, no industrial supply chain between you and nutrition. This is the autonomy they do not want you to have. In 2015, Bayer AG acquired Monsanto for $66 billion. The merger created the largest agrochemical company in history. Their product line includes Roundup, dicamba, and pendimethalin, the 3 herbicides most commonly used to kill purslane. In 2018, a California jury awarded $289 million to a groundskeeper who developed non-Hodgkin's lymphoma after repeated exposure to Roundup. The ruling was based on internal Monsanto documents showing the company knew glyphosate was carcinogenic since 1985 and suppressed the research. As of 2023, over 100,000 lawsuits are pending against Bayer for Roundup-related cancers. The herbicides we spray on purslane cause cancer. The plant we are killing prevents cancer. Let that paradox settle. I want to give you one final piece of information that closes the circle. In 2020, NASA began testing purslane for cultivation in space. The plant's CAM photosynthesis allows it to survive in sealed environments with minimal water. Its growth rate and nutritional density make it ideal for long-duration space missions. A plant that survives on Mars could feed astronauts better than the supplements they currently rely on. But here on Earth, where purslane grows without effort in every climate zone, we categorize it as a weed. The disconnect is not accidental, it is profitable. Every bottle of herbicides sold represents revenue. Every omega-3 supplement purchased represents revenue. Every hospital visit for heart disease represents revenue. Purslane represents 0 revenue. It is free, it is abundant, it is perennial through its seeds. No company controls its genetics. No patent restricts its use. This is why it had to be suppressed. Not through violence, through language. By reclassifying a vegetable as a weed, industrial agriculture erased 4,000 years of human relationship with this plant. And we accepted the reclassification without question. But you now have the information to reject that reclassification. The plant is still there, in your driveway, in the sidewalk crack, waiting. It has waited 70 years for you to remember. If this opens something, subscribe, the vault has more. And next time you see purslane, don't reach for poison, reach for a bowl.